Chapter 10 is titled Molecular Biology of the Gene. So first, scientists had to figure out what was the genetic material, what causes inheritance. They did have it narrowed down to either DNA or protein. So they performed an experiment using bacteriophages. Bacteriophages, sometimes they are for short called phages. They are viruses that only infect bacteria. The famous experiment that was performed to determine whether or not it was DNA or protein as the genetic material was performed by two scientists with the last names of Hershey and Chase. They used bacteriophages, which are types of viruses that infect bacteria, with the process of thinking these viruses will infect bacteria by injecting their genetic material. So the whole premise of this experiment was to see if the bacteriophage viruses are injecting their DNA or their protein into their host bacterial cell. So what they did is they made two different batches of these bacteriophage viruses. Batch 1, they radioactively labeled the protein. Remember, with radioactivity, you can detect it where it ends up. Batch 2, they radioactively labeled the DNA. Then what they did is they mixed these radioactively labeled phages with bacteria, and then the phages infected the bacterial cells. Step 2 is that they put this solution in a blender, agitated it, and what this did was they separated the phages outside of the bacteria from the bacterial cells. And then step 3, they centrifuged the mixture. Centrifuge basically means that they put it in this machine that spins really fast. And what it does is it makes the more dense bacteria form a pellet at the bottom of the test tube. Fourth step is to actually measure the radioactivity. Is it in the pellet or is it in the liquid? So in batch 1, when the proteins were labeled, what you can see is that the radioactivity was not in the pellets that contain the bacteria, but the radioactivity was in the liquid. The liquid represents the parts of the bacteriophage viruses that did not enter the bacterial cells. With batch 2, the radioactivity did appear in the pellet, and again, the pellet is the bacteria. So the conclusion is that DNA is the genetic material based on the fact that these bacteriophage viruses, they injected their DNA into the host bacterial cell, not the protein. In the first unit, we learned that there are macromolecules that make up cells, carbs, lipids, proteins, and then the fourth is nucleic acids. So nucleic acids are DNA and RNA. They both are made up of the same monomer called a nucleotide. However, the nucleotides differ in DNA versus RNA. What is the same about the nucleotides is that they have a phosphate group, a sugar, and a nitrogen base. RNA too, a phosphate group, a sugar, a nitrogen base. But I do see some differences. The sugar in DNA is called deoxyribosugar. The sugar in RNA is called ribosugar. They got the term deoxyribose is because if I compare this and this, ribose actually has an extra oxygen. So deoxyribose literally means without an oxygen. The other difference I see with these nucleotides is the nitrogenous bases. In DNA, the 4 types of bases are A, G, C, and T. In RNA, the 4 bases are A, G, C, and U. Aside from that, the phosphate groups are the same. Here's a closer look at all of the nitrogenous bases. You can see they're called nitrogenous because they do have nitrogen in them. So the T base, the full name is thymine. The C base is cytosine. The A base is adenine. And then the G base is guanine. RNA has these same bases except the U base is called uracil instead of thymine. There's 2 different types of nitrogenous bases, thymine and cytosine, as well as uracil. They are pyrimidines. They have a single ring structure. And then adenine and guanine are purines. They have this double ring structure. The overall 3D structure of DNA is known as a double helix, which if you look at this analogy right here, it's basically a twisted ladder. The scientists Watson and Crick are the ones that are credited with discovering this double helix structure. They discovered the structure by looking at an X-ray crystallography picture that was taken by Rosalind Franklin. So I can study the DNA structure in this 3D double helix shape, but I can also study it by visually untwisting it so that I'm studying what I call a DNA ladder. So now let's look at what parts of the nucleotide are on the sides of the ladder versus the rungs or steps. Remember, a nucleotide is one of these because it's a phosphate, a sugar, a nitrogen base. Here I see another nucleotide, a phosphate, a sugar, a nitrogen base. The sides of my ladder, those are made up of the phosphates and the sugars. Phosphate, sugar. So it's known as the sugar phosphate backbone. The bonds that hold this together are covalent bonds. Now the rungs or the steps of my ladder, they are made of nitrogen base pairs, meaning I have 2 nitrogen bases. And then those are held together by hydrogen bonds. Another scientist that contributed to the discovery of the DNA structure was Chargaff. So when he studied the structure of DNA, he noticed that the amount of A bases or adenine always matched the amount of T bases, and the amount of cytosine, C, always matched the amount of G or guanine bases. So from there, they figured out that in the DNA double helix, A always pairs with T and C always pairs with G. Also, because my purines with the double ring structure are A and G, and my pyrimidines with the single ring structure are T and C, you can see that a pyrimidine always pairs with a purine. If I had 2 purines put together, since they're the double ring structure, then those parts of the DNA ladder would be wider than when I would have 2 pyrimidines put together. Now, remember that RNA has uracil instead of thymine. So in RNA, A and U are the complementary base pairs that match up. Another aspect of DNA structure is that it exhibits anti-parallel orientation, which means that the 2 strands or the 2 sides of the DNA ladder, they line up opposite each other. So if I compare this nucleotide with this nucleotide, it looks like one's upside down because here I have the phosphate up here the phosphate is there. Now the different ends we can call the 5' end and the 3' end. Where these numbers come from is that the carbons in the sugar, the deoxyribosugar sugar, they are numbered. So carbon 1, 2, 3, 4, and then there's a carbon here, 5. So the 5' end sticks out here, and that's where a phosphate is attached. But over here, it's really the 3' end with the OH group or the hydroxyl group that's sticking out there. It is important that DNA can copy itself so that when a cell divides, the 2 cells at the end still have a complete copy of the DNA, but DNA's job really is to code for proteins. So there's this one gene, one polypeptide theory that states that one gene codes for one polypeptide. Polypeptide is a chain of amino acids. When that chain of amino acids actually folds to become a complete protein, now we can call it a protein. So the way that DNA is organized is that an organism's DNA, which all of its DNA can be called its genome, is divided up into chromosomes. Remember that prokaryotes, they just have one circular chromosome, but eukaryotes, they have multiple linear chromosomes. And then chromosomes each contain several genes. A gene, it is a segment on the chromosome that can be anywhere from 100 to 1,000 nucleotides long. One gene is going to code for one polypeptide or one protein. Now there are some proteins that do have quaternary structure, which means that it has multiple subunits. those proteins are going to require multiple genes because one gene is going to code for just one subunit of that protein. Linking this content back to our previous unit, what does it actually mean on the molecular level, the term genotype? When we wrote genotype for genetics, we wrote down the alleles. Is it big A, big A, little a? On the molecular level, a genotype really is the nucleotide sequence of that gene. If you have two different alleles, that means you're going to have two different forms of that gene, two different nucleotide sequences. Well, then phenotype, really on the molecular level, the way that you get genes being expressed is through the protein made. So phenotype is accounted for by what protein is made. Protein synthesis consists of two parts, transcription and translation. Transcription is when the DNA is rewritten into RNA. In eukaryotes, DNA does not leave the nucleus, so in eukaryotes, transcription is going to occur in the nucleus. In prokaryotes, there is no nucleus, so transcription is just occurring in the cytoplasm. Translation is when that RNA, in eukaryotes, it's actually going to leave the nucleus. It's going to go to a ribosome, and there it's going to be read in order to make a protein. Both prokaryotes and eukaryotes do have ribosomes, so translation is going to happen at the same location with prokaryotes and eukaryotes. The central dogma of biology is this flow of information from DNA to RNA to protein or polypeptide. So that's what this image is showing, just an overview where rewriting DNA to RNA is transcription, reading RNA to make a protein is translation. So if this is a long DNA molecule, and if it's part of a chromosome, and chromosomes, remember, they contain many genes. So if I look, here's gene 1, here's gene 2, here's gene 3. You can see that there's some non-coding parts of DNA that are in between genes. Here I'm looking at just one side of that DNA ladder of my gene, and you're going to see that during transcription only one side of the DNA is read. So that DNA will be read to make a corresponding RNA sequence, and then the RNA will be translated into a chain of amino acids, and then that chain of amino acids, my primary structure of my protein, that's going to fold to give me the overall tertiary structure of the protein. So now let's take a closer look at transcription. Again, it does occur in the nucleus in eukaryotes. With prokaryotes, they don't have a nucleus, so it's just going to occur in the cytoplasm, but at the DNA, wherever the DNA is. If I look at the DNA of a gene, so remember, a gene is a segment of DNA that codes for one protein. Well, it is surrounded by a region of DNA called the promoter. That's going to signal where transcription should start. And then a region of DNA called the terminator. This should signal where transcription should stop. So genes are surrounded by promoter regions and terminator regions. Initiation, that's how I'm going to have transcription start. So transcription is going to start by my transcription enzyme known as RNA polymerase. It's going to bind to the promoter region, so I can see that happening right here. And then elongation, you're going to see that this RNA polymerase, it's going to zip along the DNA, so I can see it zipping along, and it's going to make an RNA sequence. So it's called elongation because I see my RNA elongating. And then termination is when that RNA polymerase does reach the terminator region of the DNA. That's the end of the gene, so that's going to signal RNA polymerase to stop transcription, and then I have this completed RNA. Let's take a closer look at how RNA polymerase actually works. So I can see that this gray image, that is my RNA polymerase enzyme. What it's doing is it is partially unzipping my DNA. The DNA is shown in blue. And then only one strand of the DNA is going to be read to make one strand of RNA. So that's another difference between DNA and RNA, where DNA is double-stranded. Most RNA is just single-stranded. So if you look at how transcription is occurring, what the RNA polymerase is doing is if there is a G in DNA, that'll match it with a C in RNA, T in DNA, A in RNA. A in DNA, since there are no T's in RNA, it's going to match it with a U. All right, and then GC, GC. This is going to continue until there's the terminator region. There are different types of RNA. Messenger RNA, or mRNA, you'll see that that is the RNA that actually contains the message that codes for a protein. So it's really only mRNA that is translated. In eukaryotes only, mRNA goes through a specific editing process. So one thing that happens, here's my RNA transcript that's made from DNA. A cap is added to it. So what this cap actually is, it's a modified G nucleotide or guanine nucleotide, and it's added to the 5' end. And then there's also a tail that's put in. A poly A tail is what it's called because it's a bunch of A or adenine nucleotides added. There can be anywhere from 50 to 250 adenines added. What does the cap and tail do? Well, the cap and tail is going to help the mRNA do 3 things. It's going to help it exit the nucleus, which is why it makes sense that this is only occurring in eukaryotes. It helps protect the RNA from degradation by enzymes. It also helps the mRNA bind to a ribosome so it can go through translation. Now, this cap and tail, they are not translated, meaning they are not read in order to make the protein. mRNA is edited in another way, and again, this is only in eukaryotes. There's a process called splicing. Splicing is when introns are removed, exons are kept. Well, what does that actually mean? If I take a look at my DNA actually has some parts to it called introns. Introns actually do not code for a protein. It's kind of like they're intruders. The exons, those are parts of the DNA that do code for protein. That's why we can say that exons, they are expressed. During transcription, those exons and introns, those are transcribed into the RNA. We need to remove the introns because, again, they don't code for the protein. So the introns are removed, the exons are kept, and they are spliced or pasted together. Now I have my final mRNA with the introns gone. I have the cap and the tail. That is what will leave the nucleus. After transcription, translation is going to happen. Translation is the second part of protein synthesis. This occurs in the cytoplasm on a ribosome. This is the same thing in both prokaryotes and eukaryotes. So I can see in this picture that it's showing that the double-stranded DNA, only one strand is read during transcription to make a single strand of RNA. If this is the RNA that's going to be read to make a protein, it is mRNA. And I can see that the mRNA, it is read 3 bases at a time. This 3 bases is called a codon. They're called codons because one codon codes for one amino acid, which I will sometimes abbreviate as AA. Another type of RNA that helps with translation is called transfer or tRNA. It's called transfer RNA because it actually transfers amino acids to the ribosome, and it does this by matching the codon to the correct amino acid. So let's see how that happens. Here's my tRNA. All right, I can see that it is a single-stranded molecule of RNA, but it's kind of twisted up on itself. tRNAs have 2 important parts to them. They have this amino acid attachment site, and they have this 3-base anticodon. So let's say that this is my mRNA. Well, my mRNA, remember, it's read 3 bases at a time. So here's an example of a codon, the 3 bases. And the codon here is UGG on my mRNA. Well, the tRNA that's going to match up is going to carry the anticodon ACC. So the codon and the anticodon are going to be complementary. When the correct tRNA with the matching anticodon comes, it is going to bring the correct amino acid to the ribosome. There is a third type of RNA that plays a role in translation, and it is called rRNA. So if I actually look at my ribosome structure, there's two subunits. There's a large subunit and a small subunit. And I know they're just shown as blobs here, but these subunits are made of protein and another type of RNA called rRNA, which stands for ribosomal RNA. The other aspects of ribosome structure is that it has a binding site for the mRNA and binding sites for tRNA. Here's how translation is initiated. So what I'm going to have is the mRNA is first going to bind to just the small ribosomal subunit. And then the first tRNA, called the initiator tRNA, is going to come. Translation is always going to start at this start codon AUG, which means that my first tRNA molecule should have the anticodon UAC. Now, I know that this is a simplified version of a tRNA. It's not really showing the RNA structural aspect of it. It's really just showing the two important parts, the amino acid and the anticodon. So the anticodon is always associated with the amino acid methionine for the initiator tRNA. In other words, the anticodon UAC is always going to be matched up with the amino acid methionine, which here is just abbreviated MET. After that first tRNA matches, now my large ribosomal subunit is going to bind. So we just saw that the codon AUG codes for the amino acid methionine. How do we know that? Well, scientists have put together a codon chart. You are not going to have to memorize this. You will be provided one, but you do need to know how to read it. So I want you to notice that it says first base, second base, third base. So if we look to see where AUG is, well, I'm going to start here where it says first base. So A, second base in codon U. So I'm going to see where those two meet. I know I'm in this box. It's where A and U meet. For me to know which of these amino acids I use, now this third base in the codon, I'm going to see that it says UCAG. Since G is my third position, then I know I want to be here. So I'm going to go back to that box, MET or methionine. That's what AUG codes for. Translation elongation is how do I keep adding amino acids to my protein? How do I elongate my amino acid chain? So it's really the same process where it starts with codon recognition. Remember, the three bases on the mRNA are called the codon. The three bases on the tRNA are called the anticodon. So the correct tRNA is going to come in by matching its anticodon to the codon. So now I have my tRNA, and then I'm going to have a peptide bond formed between the amino acids. Remember, a peptide bond is the name of the covalent bond between amino acids. So I'm going to have a peptide bond formed between this amino acid and then the amino acids that have already been built. This tRNA now, it's empty. It gave up its amino acid. So the empty tRNA will leave. It can be used again. It'll just pick up a new amino acid. And now translocation means that this ribosome is going to shift over. And now I'm going to have an empty tRNA site, and the whole process is going to continue, where I'm going to have anticodon of tRNA match up with codon of mRNA. It's going to bring the correct amino acid, and then I'm going to be joining these amino acids to build my protein. Translation is eventually going to be terminated. Before we get to that, this is a summary of everything we have seen, putting together transcription and translation. So remember, transcription is when mRNA is transcribed from a DNA template using RNA polymerase. And now to prepare for translation, amino acids are going to be attached to their specific tRNA. An enzyme is going to do that with the help of ATP. And then you're going to start translation really at that start codon. elongation, I'm going to continue having tRNAs come and bring the proper amino acid. Those amino acids will be put together to make my protein. And then finally, there is a stop codon. So there are some stop codons, which really are signals that tell the ribosome to stop making the protein. So at this point, the protein or the polypeptide will be released. Stop codons, they really are just signals. They do not code for a specific amino acid. I know everyone thinks that DNA, that's really what makes us who we are, but in reality, DNA is the instruction for proteins, and we really are a result of the proteins we make. So if there is a change in the DNA called a mutation, that can change an aspect of our trait by changing the protein by this trickle-down effect. If there is a change in the DNA, well, that will change the mRNA made, which could change one or more amino acids and then change the overall protein. And it really is that overall 3D structure of the protein that matters. While mutations do have a negative connotation, all mutations are not harmful. Some can be silent, some could actually be beneficial, or it could just be a neutral change that really doesn't affect us at all. So let's go through an example by seeing what causes sickle cell disease. Here is just one section of the normal hemoglobin DNA. There's CTT. Well, then that part of the DNA is going to be transcribed into the mRNA GAA, and then this mRNA codon codes for the glutamate amino acid. So this sequence of the DNA is associated with the allele big S. Sickle cell disease, it is caused by a recessive allele. Well, remember what makes alleles different, I told you, is the nucleotide sequence. So here is the changed or mutated version of the hemoglobin DNA that causes sickle cell. Instead of a T, there's an A. So the CAT here is going to be transcribed into the mRNA GUA, and this codon actually codes for a different amino acid, valine. This one change in the amino acid is going to change the overall structure of the protein, and it's going to cause sickle cell. There are different types of mutations. I'm calling all of these single nucleotide mutations just to differentiate from the mutations we learned about in the last unit, those larger scale chromosomal mutations where if you have a large chunk of a chromosome deleted, duplicated, inverted, or a translocation, I'm actually talking about several nucleotides, possibly even several genes. But here I'm just talking about one nucleotide. A point mutation is when one nucleotide is substituted or replaced for another. These are called point mutations because they just can possibly change the protein at a single point by changing just one amino acid. This is different than the other type of mutations called frameshift, and you'll see what I mean. There are 3 different types of point mutations. The first is silent. Silent is when the changed codon codes for the same amino acid. Remember that mutations, they are changes in DNA, but DNA changes are going to cause a change in mRNA, which means the codons will change. So let's say the original codon is UGU. If I looked on the codon chart, that codes for the amino acid cis or cysteine. If there's a mutation, now the new codon is actually UGC, there's a lot of repetition on that codon chart. UGC also codes for cysteine. So this is a silent mutation in the fact that my protein will not change because I'm not changing any of the amino acids of that protein. Second type of point mutation is called a missense. This is when the changed codon codes for a different amino acid. So if the original codon was UGU and coded for cysteine, if that third base changed to a G and now my codon is UGG, you will see that that codes for tryptophan. So this is a missense. I'm changing one amino acid into another. The third type of point mutation is called a nonsense. Nonsense point mutation is when the changed codon now codes for a stop codon instead of amino acids. So with this, the protein is going to be stopped short. If the original codon was UGU that coded for the amino acid cysteine, and now the mutated codon is UGA, you will actually see on the codon chart that this is a stop codon. The other type of mutations aside from point mutations are frame shift mutations. Frame shift mutations can also be called insertions or deletions because this is when nucleotides are either inserted or deleted. Point mutations, there is just a substitution. So you can see with frame shift, I'm actually changing the amount of nucleotides at my gene. And because of this, I'm going to alter that reading frame, that triplet grouping, the 3x3 reading frame of the mRNA. So if you look here, if this is my original mRNA that was coded for by my DNA, then here's my original protein, the amino acid chain. If there is a deletion in the DNA, there will also be a deletion in the RNA. mRNA is always read 3 bases at a time. So if this is deleted, all of these basically shift over this way, and now every amino acid after my mutation is going to be different. All right, so I can see that these amino acids are different. If you can imagine, the same thing is going to happen with an insertion. Let's say an extra base is inserted here. Well, that's going to shift everything over this way now. And again, every amino acid after the mutation will be different. So how do mutations occur? Source of mutations, this is called mutagenesis. There are some that are referred to as spontaneous mutations. This is when there's errors during DNA replication or errors during crossing over of meiosis. Even though DNA polymerases do have this proofreading or spell check mechanism, because the process is happening so fast, there can still be errors during DNA replication. There can also be physical or chemical agents called mutagens that can change the DNA. Example of physical mutagens are high energy radiation that can damage DNA, such as X-ray radiation, UV radiation. There's also certain chemicals that what they do is they mimic the bases of DNA, and during DNA replication, they get inserted instead of the actual nucleotides, and this changes the DNA.